welcome to Let's Meet the Virologist, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with students, postdocs, and other virologists so that you can learn who they are and what they do. I am Larissa Thackeray, and I'm hosting this podcast from America's Heartland in St. Louis, Missouri. On June 21st, 2022, we talked with Joanna Stuba-Solinska, an assistant professor at Auburn University. She received her first master's degree in Poland and a second master's and PhD at Northern Illinois University. Her postdoctoral training was at the National Institute of Health, National Cancer Institute. Currently, her lab focuses on understanding coding and non-coding viral RNA, structure, function, and regulation. All right, well, thanks for talking with us today. Why don't you tell us a little bit about yourself?
Hello, my name is Joanna Stuba-Solinska. I am an assistant professor in the Department of Biological Sciences at Auburn University. I come from Poland, from Bydgoszcz, Poland. I came to the United States 17 years ago. And since then, I've been studying viruses with passion, and to be exact, RNA of the viruses, their structure, function, and regulation.
Great. And can you tell us sort of back when you were younger, when did you first become interested in science and then virology?
Interested in science, I was always interested in science. I recall going through my grandma's books of human anatomy and scribbling on them. So I remember that it's been a passion of mine since the beginning. With viruses, being interested in viruses, I recall that sometime in high school, I went to the library and I found this book about HIV. That was the only book that I could find about viruses. And I remember being absolutely fascinated by the complex structure of the virus and the fact that it's not really alive, yet it behaves kind of like alive. And it can be such a devastating, entity causing the disease that basically takes off somebody's life. I remember that happened in high school.
Cool. And can you describe then a little bit your journey through sort of your undergrad, graduate, postdoc, sort of where did you go? How did you choose those places? That kind of a thing.
So I received my first master's degree in biotechnology in my home city in Bydgoszcz, Poland. And right after that, I applied to graduate school in the United States, and I did my second master's degree and PhD at Northern Illinois University. After that, I started applying for postdoctoral positions all over the United States, and I was lucky to find a position at NIH, NCI. And this is where I did my first postdoctoral fellow position, and that was followed by research fellow position. And finally, in the end, after more or less 5 years, I was offered a staff scientist position at NIH. I studied viral RNA, its structure and function using different viral systems from HIV actually to dengue viruses. I was lucky to have a collaboration with NIH, NIAID, and I received a training and certification to work in BSL-4. There I worked with Ebola, chikungunya, and other selective agents. And after about 6 years being in the DC area, so to speak, I applied to academic positions, and basically I received an opportunity to become an assistant professor at Auburn University.
Okay, great. And then can you tell us then a little bit about your lab? I guess sort of some of the big picture sort of questions you're trying to answer, and then maybe some of the techniques that you use in your lab to sort of address those questions.
Okay, so we are studying a viral RNA, coding and uncoding RNA, how its structure pertains to its function, and how it is regulated by post-transcriptional or epitranscriptomic modifications. The techniques that we usually use in my lab is SHAPE and SHAPE-MAP, which is biochemical probing technique that allows to solve the secondary structure of the RNA inside the cells, inside the virions, also as an in vitro transcript. Once we have those structures, we want to basically ask a question, which of these structures are important for viral RNA and for the virus itself, for its infectivity, for progeny virion production, for replication, and so on. Epitranscriptomic modifications are sort of an additional layer to that story. They make the RNA even more complex than it really is, because they can actually completely change the structure of the RNA. They can affect the interactions of the RNA with other RNAs, with other proteins. So we are focusing on this very abundant but very mysterious modification that is called a pseudouridine that has been discovered 7 decades ago, but not much is known about that particular modification, especially in the context of viral RNA. So we found that one of the RNAs that we are interested in is decorated with that modification, and we want to not only identify where it is, how frequently it occurs, but also what it does. And that's what it does is the most sort of difficult aspect to address, because those modifications, they are installed by cellular machinery, and oftentimes people kind of question whether they're not sort of a side effect of the virus being in a cellular environment. So proving their functionality is the key to basically address their importance for the viral replication.
Right. And can you kind of maybe speak a little bit more generally about how RNA viruses in particular, because they have sort of limited coding capacity, why they have to use their RNA not just for translation, but for many of these other functions that you're talking about? Can you talk a little bit about that?
Right. Viruses, that's one of the reasons why we actually study viral RNA, because it's so complex. It needs to accommodate all these functions from replication, transcription, translation. And because of its limited coding capacity, it has to do it in kind of intricate ways. And that involves basically folding in these complex structure, complex motifs that on top of its secondary and tertiary fold can be regulated by these epitranscriptomic modifications. And many viruses use these tricky strategies to expand their coding capacity from, let's say, folding into a pseudoknot structures that regulate expression of one open reading frame versus the other one, regulating the ratio of those proteins expression. They use these very complex structures to support recombination, for example. Oftentimes these structures are sort of hotspots of recombination. That was actually a topic of my PhD. That's why I'm kind of passionate about that. So truly the structure of the viral RNA is something that dictates the capacity, much larger capacity to code and to function.
Right. And then can you talk a little bit about why that presents challenges, as you were saying, to try and identify precisely what a pseudoknot or a particular modification actually does? Because there are often 3 or 4 things that nucleotide in a way could be doing. How do you actually show that, that particular modification or that particular nucleotide is important for one thing without affecting all the others?
Indeed, because of these modifications and structures are oftentimes regulated by cellular enzymes, it is not easy to address their functionality. Imagine this. Naturally, if you want to study a given nucleotide or its modification, and you are lucky and you identify an enzyme that is responsible for that modification, the very first thing you would do is basically knock out or knock down that enzyme. But those enzymes, they oftentimes have other functionalities, not just that catalytic modification that I install, but sometimes they associate with a given RNA and do completely different, stabilize that RNA, support its folding. On top of that, if you knock down that particular enzyme, you will affect other cellular processes. So the effect you will see might not be just the effect of the knocking down that particular modification, but also knocking down the enzyme that affects other cellular important processes. So to compensate for that pleiotropic effect that you may see, you have to also, let's say, modify a given residue. But oops, then we are getting into changing one nucleotide into another nucleotide, which will affect the structure of the RNA. So truly, again, it's a question of, did we actually remove a given modification, or did we affect the entire structure of the RNA, therefore its function? So oftentimes we have to have at least two approaches like that in order to get to that consensus answer. It is not easy task to accomplish.
I see. So you try and get basically a couple of techniques to agree on the answer, to kind of say, with the totality of this, we think that this is the function of the.
Absolutely. You have to approach it from the perspective of the RNA itself, but also from that cellular enzymatic approach in order to find a consensus answer.
And then can you discuss a little bit whether understanding sort of these different modifications or functions, does that tell us anything about how RNA functions, like the host RNA? Does it actually give insight into other sort of cellular processes that are actually going on in mammalian or other hosts?
Absolutely. So there are many aspects of that. Oftentimes when viruses infect the cell, they not only decorate their RNA with these epitranscriptomic modifications to kind of hide themselves from the cellular immune response, but also they dysregulate the cellular processes to sort of seal these enzymes. So that oftentimes results in lowering of epitranscriptomic modifications on other cellular messenger RNAs. So viral infection truly kind of throws a kind of a wreck in the cellular environment. And epitranscriptomic processes is one of the things that are completely dysregulated. Viruses kind of usurp those mechanisms to hide from the immune response, but at the same time, they kind of destroy the homeostasis that is there as basically a consequence.
Right. And can you tell us then a little bit about your lab? So how long have you been an assistant professor? What has the last 2 years been like trying to be doing research? Do you have graduate students, postdocs? Like who are the people that are in your lab? How's that going?
So I've been assistant professor at Auburn University for 5 years. I currently have 2 PhD students and a postdoctoral fellow and a bunch of undergraduate students that help with different aspects of graduate students and postdocs proposals. The last 2 years have not been that great because of obviously a pandemic that affected quite a bit the progress of the research. Unfortunately, some of my students were sick. Actually, one of my students literally got diagnosed with COVID last week. So it is an ongoing pandemic. Nothing has changed. We are all, of course, vaccinated, boosted, and all of that, but we still get sick. Thankfully, we don't end up in a hospital. So last 2 years were difficult, but we are continuing our research despite the issues. The most, I would say, detrimental aspect of the pandemic was the fact that we couldn't get in person with other scientists to exchange the knowledge and skill, to basically exchange ideas, to look into other people's research and share our research. So having an opportunity to finally participate in an in-person meeting is absolutely critical for students, for postdocs. Networking is important for assistant professor to build in those connections, collaborations. Nothing happens in just one lab. Nowadays, we need those collaborations. We need to talk with each other. And unfortunately, pandemic hindered that quite a bit. The good thing is that we got more accustomed to online meetings like Zoom and MS Teams and other resources, so that social media sort of kept us going. but there is nothing like in-person meeting. Nothing is going to replace that. That's for sure.
Right, right. And speaking of that, I believe you're talking at the Satellite Symposium. So could you just give us a brief overview of what you're going to be talking about?
I will be actually talking about the pseudouridine modification of the long non-coding RNA coded by Kaposi sarcoma-associated herpes virus. I will be talking about its localization, stoichiometry, and the ways we want to apply to resolve its functionality in the context of that particular RNA biology, but also in the context of viral infection. So that will be a main subject of my talk.
Great. And could you tell us a little bit about Kaposi sarcoma virus? So I think that's not, I don't think I've interviewed someone that actually works on that virus. So can you talk a little bit about it?
So this is a DNA virus. It's an oncogenic virus that causes different kinds of malignancies in immunocompromised people, especially HIV-infected people. If anybody have ever seen an HIV-infected person sort of developing AIDS disease, at some point in the patient's life, you can develop these sarcomas on the skin, like a darker patches. This is a result of actually a secondary infection with Kaposi sarcoma-associated herpes virus. KSHV is sort of an endemic virus in Mediterranean regions. It's actually responsible for about 50% of cancers in sub-Saharan Africa. It is also causing quite prevalent infections in people after transplant, so basically immunocompromised people. This particular virus, just like all herpes viruses, is absolutely fascinating because herpes viruses have been evolving with people since the beginning. They are the most sophisticated viruses ever because basically they infect us, stay with us for the rest of our life, and we are absolutely fine most of the time. And sometimes when triggered by stress or by some unknown conditions, they can reemerge and cause the disease. So they are very interesting viruses to study. They have two phases of infections: latent, when they stay quiet. They basically hide in nerves and they kind of survive the immune response with their microRNAs or long non-coding RNAs. And once they sort of know that the cell is kind of weakened, then they go into a lytic reactivation, and that results in a replication and progeny virion production and spread. So they're kind of a smart viral system. They're also, and their genome is also quite big, so they're very difficult to manipulate and study. They have lots of genes. Those genes oftentimes overlap. The transcripts are produced in sense and anti-sense orientation. They're spliced, so it's not an easy system to study, but that's why it's so fascinating.
And the work that you're describing, what are some of the major techniques that you're using? Is this like the SHAPE or that you were discussing before, or are you using other techniques?
Oh, we use a plethora of techniques. So SHAPE would address just the secondary structure of a given RNA, but like I said, there is another layer to it, which is epitranscriptomic modifications. These we mapped with next generation sequencing techniques and chemical modifications of the RNA that basically cause the reverse transcriptase to stop at the site of modification, and that allows us to identify those sites. We are also studying RNA protein interactions, which pertain to their function, that we use. We basically use a method that is called RNA antisense purification with mass spectrometry, and then traditional molecular biology techniques from Western blots, Northern blots, RT reaction, PCR, quantitative PCR. So all the typical molecular biology techniques that run labs forever.
Right, right. And then I guess just looking into sort of the future, what are your plans sort of like in the medium term, long term? What are you interested in studying?
Mm-hmm. So in general, I've been always fascinated with not only a structure of the RNA, but its modification. But pseudouridine is just one of about 170 of those. And what's more, pseudouridines can be modified even further into a methyl pseudouridines and more. I would be interested to not only address the functionality of that particular modification, but also how that particular modification cooperates or collaborates with other modifications. They all exist at the same time. They maybe not. Maybe one outcompetes the other one. Maybe they are sort of antagonistic. Maybe they actually are synergistic and they affect the RNA and the viral infectivity cycle in a different way. These are questions that are not addressed really in the field. Also, knowing what is the fraction of a modification. And we are talking about one single transcript, but truly there are millions copies of that transcript. Some of them are modified, some of them are not. And lastly, what reads these modifications? There are no known pseudouridine readers or proteins that recognize, bind a pseudouridine and fulfill its function. Like with M6A modifications, which is another very abundant modification, we know those readers, writers, and eraser. For pseudouridine, nothing is known. So it's been so abundant, it's actually second most abundant after M6A, and nothing is known. So I want to address a global pseudouridinylation regulation of the RNA and viral infectivity by pseudouridinylation.
Great. Well, thanks so much for talking with us today. And we look forward to hearing about your work at ASV. Sounds very interesting. And thanks for talking with us.
Thank you again.
This has been Let's Meet the Virologists, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, and other podcast providers, or at lmtv.podbean.com.